Hello, and welcome to Science with Mr. Morgan. In today's episode, we're going to talk about molecular genetics and pedigrees. In a previous episode, we dove into Mendelian genetics, but now let's take a look at modern genetics. Mendel laid the groundwork for modern genetics, but his work left kind of a few of unanswered questions. Two such questions include what exactly are dominant and recessive factors that determine how all organisms look, and how do they work? Since Mendel's time, we have learned a lot more about genetics, which helps to provide answers to those questions. Genetic material is made out of DNA, and DNA makes up the hereditary factors that Mendel identified. Therefore, genetic material is the hereditary factors. If you have seen the lesson on DNA, you know that DNA is wound into chromosomes. Each chromosome contains a long chain of DNA that encodes for hundreds or thousands of genes, and each of these genes vary slightly from individual to individual, which makes you different from me. These variations are known as alleles, which are what Mendel referred to as factors. These alleles lead us to a conversation about genotype versus phenotype. So first up, what is a genotype? It is the way to describe a combination of alleles that an individual has for a certain gene. For each gene, an organism has two alleles. They have one from each chromosome of a homologous pair of chromosomes, aka one from mom and one from dad. This is represented by letter combinations like capital T, capital T, capital T, lowercase t, or lowercase t, lowercase t. These combinations are known as homozygous or heterozygous. Now, homozygous have two variations, which are homozygous dominant or homozygous recessive. In general, homozygous means two of the same allele. For example, capital T, or lowercase t, lowercase t. Homozygous dominant means two dominant alleles like capital T, capital T. Homozygous recessive means two recessive alleles like lowercase t, lowercase t. Heterozygous means one dominant allele and one recessive allele like capital T, lowercase t. Now, a good way to remember what a genotype is that it is what is on the inside of you, not what you see. And now that we've looked at genotypes, let's look at phenotypes. So phenotypes are the way to describe the traits that you see. And these traits are expressed by the genotype. Now, that being said, organisms with different genotypes can still have the same phenotype. So your genetics can be different, but they can be physically seen the same way. Mendel's pea plants had the genotypes of capital T, and capital T, lowercase t, but each one was tall. So being tall is the phenotype. A good way to remember what a phenotype is that it is what you see, not what is on the inside. Next up, let's talk about pedigrees. So what is a pedigree? Essentially, it's just a chart that shows the inheritance of a trait over several generations. These are commonly created for families, and it outlines the inheritance patterns of genetic disorders and traits. It can also be used for things such as cows if you're a cattle farmer. They can be used to determine if a trait is sex-linked or autosomal. If a trait is sex-linked, it means that it is either on the X or the Y chromosomes, aka the sex chromosomes. If a trait is autosomal, it means that the trait is on a chromosome that does not determine sex, aka any of the other chromosomes that are not the X or Y chromosome. Pedigrees can be used to determine if the trait is inherited in a dominant or a recessive fashion. They can also help predict the probability that offspring will inherit a genetic disorder. When you're analyzing a pedigree, there are certain things that you need to keep in mind. With autosomal recessive inheritance, all affected individuals will be homozygous recessive. For example, they would all have lowercase t for their alleles. With dominant inheritance, all affected individuals will have at least one dominant allele. This means they will either be homozygous dominant, like capital T, or heterozygous, like capital T, lowercase t. But with a capital T there, the dominant allele is expressed. With sex-linked inheritance, more males, aka those with XY chromosomes, than females, aka those with XX chromosomes, will carry the trait. This type of inheritance is usually recessive, which is why more males are affected. Males only have one X chromosome, which means that if the allele is present, it will be expressed. while females have two X chromosomes, which means that the allele would need to be present on both of the X chromosomes in order to be expressed. Continuing this conversation about sex-linked inheritance, what determines the sex of a baby? If you said chromosomes, you'd be correct. Remember, there are 23 pairs of chromosomes in humans, aka 46 total, and one of those pairs are the sex chromosomes. Now, everyone has at least two sex chromosomes. And I know I said at least, but most people only have two. We will talk a little bit about examples where individuals have more than two in the genetic disorders and mutations episode. As I mentioned before, these sex chromosomes can either be an X or a Y chromosome. And as I said a few seconds ago, females have XX chromosome and males have XY chromosome. And I'm referring to the biological sex of an individual here. The allele present on the sperm is what determines the biological sex of the offspring. Remember, sperm are haploid, so they will only have one sex chromosome present, which will either be an X or a Y, as well as all of the other chromosomes. But they carry 23 chromosomes total because they carry half. Eggs are also haploid, but since females have homozygous X alleles, all eggs have X chromosomes. This is why sperm cells determine the sex of the baby, because eggs will always provide an X, but sperm cells will either provide an X or a Y. Now that we've spent time talking about sex-linked inheritance, what is it specifically? By definition, it is the inheritance of traits that are located on genes on the sex chromosomes. Now, this is a special pattern of inheritance that doesn't fit Mendel's rules and typically refers to traits due to genes on the X chromosomes, but that's not always the case. A specific example of an X-linked recessive condition is red-green colorblindness. People with this type of colorblindness cannot tell the difference between red and green, meaning they often see shades of brown instead of either one of those colors. And I know that we briefly touched on this, but it predominantly affects males more than females. And again, this is because males only have one X chromosome, so if the allele is present, that male will be colorblind. Since females have two X chromosomes, they would need that allele to be present on both chromosomes for it to be expressed. Females that have one dominant allele and one recessive allele are referred to as carriers. And carriers carry the allele and can pass it on to their offspring, but they do not express it. Now, sex-linked inheritance is not the only non-Mendelian inheritance. And this refers to the inheritance of traits that is not always as simple as Mendel's rules. Mendel's characteristics were controlled by one gene that had only two possible alleles. And in this case, one was completely dominant over the other. Now, there are various exceptions that typically have something to do with the dominant allele itself. These exceptions include incomplete dominance, codominance, and polygenic traits. First up, let's look at incomplete dominance. This is when one allele is not always completely dominant over another one. And this can lead to a phenotype that is somewhere between the two parents. Let's look at an example of incomplete dominance by looking at flower color. In this example, we're going to crossbreed a red snapdragon, represented by capital R, with a white snapdragon, represented with a lowercase r. In this example, this crossbreed will result in a pink snapdragon, represented by capital R, lowercase r. Now, that was flowers, but not related to people. So does incomplete dominance impact people too? Yes. Let's look at an example by talking about sickle cell anemia. This is a disease in which the blood protein hemoglobin is produced incorrectly, which makes the red blood cells have a sickle shape. This shape causes the affected red blood cells to struggle to pass through very small blood vessels. Capillaries, if you know your blood vessels. A person who is homozygous recessive, which is represented by a lowercase s, will have sickle cells. And a person who is homozygous dominant, which is represented by capital S, will have normal red blood cells. A person who is heterozygous, which is represented by capital S, lowercase s, will have some misshapen cells and some normal cells. Another type of non-Mendelian inheritance is codominance. In codominance, there are two alleles for the same gene that are expressed equally in the phenotype. An example of this includes blood type. Now, normally, genes only have two alleles, but blood type is a little weird where it has three alleles instead of the normal two. Blood types can be A, B, AB, or O. And these types are named for the protein that are attached to the outside of the blood cell. Blood type A, represented with capital I, capital A, and B, represented with capital I, capital B, are dominant, while O, represented with a lowercase I, is recessive. Capital I, capital A codes for the A antigen. Capital I, capital B codes for the B antigen. And lowercase I codes for no antigen. People with type A blood can be homozygous, which is represented with capital I, capital A, capital I, capital A, or heterozygous, which is represented by capital I, capital A, lowercase I. People with type B blood can be homozygous, which is represented by capital I, capital B, capital I, capital B, or heterozygous, which is represented by capital I, capital B, lowercase I. People with type AB blood are heterozygous, which is represented by capital I, capital A, capital I, capital B. And both alleles are expressed completely, which is different than Mendel's rule of inheritance, where one allele is always expressed over top of the other. People with type O blood are homozygous recessive, which is represented by lowercase I, lowercase I. The last example of non-Mendelian inheritance is polygenic traits, or at least the last example we're going to talk about. So what is polygenic inheritance? This occurs when a trait is controlled by more than one gene. Now, traits are typically polygenic when there is wide variation in the trait. Examples of this include height, skin color, hair color, eye color. When you analyze the population, these traits often result in a bell-shaped curve. Now, most people fall in the middle of the phenotypic range. For example, average height, while very few are at the extremes, either really tall or really short. At one end of the bell curve will be individuals who are recessive for all of the alleles, which could be represented by a lowercase a, lowercase a, lowercase b, lowercase b, lowercase c, lowercase c. And at the other end will be individuals that are dominant for the alleles, which could be represented by capital A, capital A, capital B, capital B, capital C, capital C. The middle of the curve will be for individuals who have a combination of dominant and recessive alleles, which could be represented by something like capital A, lowercase a, capital B, lowercase b, capital C, lowercase c, or capital A, lowercase a, capital B, lowercase c, lowercase c. And I know that was complicated, but that brings us to the end of today's lesson. Until next time.